Hello, folks, and welcome to the last two sections of our notes on gene expression regulation for Unit 6. In this one, we'll be doing Part 4, Mutations and Biotechnology. So, Mutations and Biotechnology 6.7 and 6.8. As you can see in the book, mutations is kind of scattered all over the place. And biotechnology is Chapter 20. We're not going into as much detail as Chapter 20 does, so whether you read that or not is up to you, depending how much you want to know about it. So, mutations. Things change. Let's start off with types of mutations. Okay. Mutations are changes in the genetic material of a virus or cell. I think you guys all know that. A point mutation is a chemical change in just one base pair of a gene. So instead of a gene being AATTGCGCA, it might be the same thing, but replace that last A with a G. That's a point mutation. Um, and that can lead to the production of an abnormal protein. If a mutation has an adverse or negative effect on the phenotype of an organism, the condition is referred to as a genetic disorder or hereditary disease. Here we can see one for hemoglobin. In normal hemoglobin, we got this codon sequence in that gene, which leads to this mRNA, which gives you a glutamine right here. Over here, we do a point mutation. We switch out the T for an A, which winds up in the end changing just one amino acid in the hemoglobin from glutamine to valine, and that is what causes sickling. So, for a relatively minor change, a big effect. Types of small scale mutations. Let's start off, and those are generally point mutations. Within a gene can be divided into two general categories. We have nucleotide pair substitutions, where we switch out one nucleotide for another, switch the A for a G, a C, or a T, or where we do insertions or deletions, putting in extra nucleotides or getting rid of some nucleotides altogether, shortening it all up. Substitutions replace one nucleotide and its partner with another pair of nucleotides. They can be everywhere from make no difference at all to being pretty devastating. So, a silent mutation is one that has no effect on the amino acid produced by a codon because remember, codons have that redundancy. So we could change GAA to GAG, not going to make a difference in the final protein or phenotype because they both code for glutamine. On the other hand, we have what we call missense mutations, where they still code for amino acid, okay, but not the correct amino acid, like we saw in sickle cell. If we change GAC, that's aspartamine, but if it's GAA, now it's glutamine. And that may have no effect. It may have a minor negative effect or a minor positive effect too, but it or it could have a major effect depending on what's going on. But these ones are relatively small changes, and they can have an effect. Nonsense mutations change an amino acid codon into a stop codon, okay, which nearly always leads to a non-functional protein. So let's see some images of that. So here's our original DNA strand, and it codes for, in the end, these four amino acids and then a stop. Okay. If we do a silent substitution mutation, put an A instead of a G there, we wind up still getting glycine in our last position. So as you can see, the polypeptide doesn't change at all. Same top one here, but now we do a T instead of a C here. We change it from a glycine to a serine. So that's a missense mutation. Got potential to make some changes. And here is a nonsense one. We change this A, I put an A here instead of a T, turns this one into a stop codon, which means we just cut that thing short and it's definitely not going to work. Now, those are point mutations where we do substitution, switch things in or out. Insertions and deletions, we actually insert extra ones or we take out some. So we're either making that gene longer or shorter. And these mutations generally have disastrous effects on the resulting protein way more often than substitutions do. Substitutions are less likely to have dramatic effects. Insertions and deletions are very likely to have disastrous effects on the functionality of that protein. Insertion and deletion nucleotides may alter what we call the reading frame and cause a frame shift mutation, and that'll be easier to discuss with a visual. So let's get some up here. So here is an insertion. We just add an extra A in here. You notice we're a little bit longer on this end, and that extra A in there makes it a stop codon. So we got to a total nonsense gene. Okay, so that's pretty bad. You're not going to make that protein at all. All right. Now, here's one where we get a frame shift. We did a deletion here. We got rid of that A. Okay, and so that gets rid of that U, which means instead of up here where we read UUU, and then for phenylalanine, and then GGC for glycine, now we read UUG because this G came down over to here now. So UUG, which codes for leucine, and then it's GCU because that U went over here, which codes for alanine, and then we don't have a stop codon over here anymore. So after an insertion or deletion, most of the time, if it's not a stop codon, we get a frame shift and everything shifts down one or up one. And since there's no like spaces or anything like we do when we're writing a word like cat space, dog space, that helps us separate it. So if it was CA space DOG, we'd still know it was probably cat than dog. Here, mRNA has, and the tRNAs and the ribosomes have no way of knowing that. They just read it three, then the next three, then the next three. So we shift it down and we get leucine, alanine, and then that polypeptide just keeps on going. And here's a kind of a showing all of them here together, so you can take a look at that if you want to and pause the video. So let's start talking about how mutations affect organisms, other than things like cancer and genetic diseases and things. So new mutations and mutagens. Spontaneous mutations can occur during DNA replication. When you're making copies of the DNA, you can just make a mistake. It doesn't happen often, and we often repair it, but it can happen. We can get mutations happen when we do recombination, when we're going through the process of meiosis, or when we're doing repair, we can make mistakes in the repair as well. So that can happen spontaneously with no outside influence. We can also get mutations caused by things called mutagens, which are either physical or chemical agents that can cause mutation. So UV radiation, tobacco smoke, asbestos fibers, you name it, things you think of as carcinogens, those are mutagens. Let's review how genes determine phenotype here because it plays into how mutations work. So genes code for polypeptides or RNA. Those polypeptides or RNA then carry out their role in the cell, which affects how that cell acts, thus affecting the overall phenotype, whether you got blue eyes or you're lactose intolerant or you've got a properly shaped hemoglobin molecule or what have you. The function and amount of the gene products determine the phenotype of organisms. So not just what that gene product, RNA or polypeptide does, but how much of it you have. If you have more of it, you're going to have more of that. So it's kind of what works with genetics. We talked about like if you got two dominant alleles or a dominant and a recessive allele, right? The two dominant alleles are going to produce more melanin pigment and make your skin or hair tones darker than if you have just one or none, right? So the more of those dominant alleles you have, the darker the skin tone or the hair color. So function of the product and the amount of the product determines phenotype. The normal function of genes and their products collectively comprises the normal function of organisms. All these genes and their products working together properly, you get the normal function. Okay? If you disrupt those genes and thus their products, we can get new phenotypes. Now, sometimes those new phenotypes are things like genetic diseases and cancer. But are they always bad? Maybe. It kind of all depends. Some of them obviously are bad because they will cause you to not even develop in the womb, and you'll be spontaneous abortions and things of that nature, stillbirths. They'll cause genetic diseases that definitely make it harder for you to survive and have a rougher life. Or they can cause cancer and all that kind of stuff. But for the ones that don't do those types of things, it kind of all depends on how much effect they have on the function and the product. So how much do the mutation affect the polypeptide or the RNA and the environmental context? Think back to evolution. Okay, we're all talking about survival of the fittest, right? If that change in your phenotype, if you get a change, if it's a major change or a minor change, that's the how much effect. If it helps you survive and or reproduce, that's actually a good mutation. If it makes it harder for you to survive or reproduce, it's a bad mutation. If it doesn't really make any difference, which happens with mutations, especially silent ones, obviously, no big deal. If you recall from regular biology, we talked about The rock pocket mice, okay, in the Southwest. Um, Normally, when they're living on when they're light colored and they live on light colored rock, and that's great. Hard for the predators to see them. So if you're light colored, you're camouflaged, you're going to live longer. Every once in a while, there is a mutant that produces a dark color. If they're living in a light colored environment, they're a lot less likely to survive long enough to pass that on to their kids, which is why the population stays mostly light. On the other hand, if they're living on these lava fields with dark rock, which one's got a better chance of survival? Yeah, the one with the dark mutation. Light mutations stand out like a sore thumb. So both of these mutations or variations, neither is bad or good on its own. What makes it bad or good is the environmental conditions in which they are living. So mutations are the primary source of genetic variation. They're what driving evolution um, in context with changes in the environment. So you got new variations, changes in the environment, put those together, you got evolution. All right, here we can see the effect of one of those mutagens we talked about just in the last slide, which would be UV light from sunlight. So a segment of a polynucleotide containing two adjacent thymine bases in this um, example can come in here and it can mess with the thymines binding across here, and it can form a dimer between those two thymines, getting them to covalently bond to each other here. Because normally we don't have that. There's no covalent bonding between the different rungs of the ladder. And that, as you can see here, causes a kink in the DNA, which will mess with replication and transcription. So those dimers that can be formed by pyrimidines due to exposure to UV light um, can cause some of these mutations that we're talking about. All right, there are some reproductive processes that can increase the amount of variation um, within organisms and that can allow organisms to share those variations. And we call that genetic recombination. Genetic recombination is the combining of DNA from two sources, and that can happen naturally in certain organisms, particularly prokaryotes, and it contributes to diversity because normally prokaryotes are just clones, but they do have the ability to move DNA from different individuals from one to the other through processes of transformation, transduction, and conjugation. We call this horizontal gene transfer. It's not something we see in eukaryotes because, and ones that produce sexually, there's no real need for it, and there's no good mechanism for it. Um, we can take these techniques, or some of them, and modify them in genetic engineering and biotechnology to do recombinant DNA and transfer genes from one organism into another. We'll talk about that a little bit later, but let's take a look at each of these just a little bit closer. So, in the process of transformation, a prokaryotic cell can take up and incorporate foreign DNA from the surrounding environment in a process called transformation. And takes up what we call naked DNA. So, here is our bacterial cell. We've got some naked DNA floating around outside. These are plasmids, right? So, a circle of DNA from another bacterium, most likely, containing a gene, could be a gene for antibiotic resistance, and it can take it up. And now we've added a new gene to this bacteria that wasn't there before. So a new phenotype, a new polypeptide, something that may help it survive. That's transformation. And we do use that in biotech. And we'll talk about that a little bit more when we get to biotech. Next, transduction is the movement of genes between bacteria by bacteriophages. Bacteriophages are viruses that infect bacteria. So obviously, what do viruses do? They inject their genetic material into their host. Okay. So this is a way that we can get changes to the bacterial DNA via the injection of it of viruses. And we have used this at times in biotech and genetic engineering to inject the DNA or RNA that we want if we're doing a retrovirus bacteriophage into bacteria or into our own tissues or things like that. We can infect it with a virus that will actually insert the DNA we want. And then conjugation in plasmids. So sometimes bacteria, rather than just taking up random ones from the environment or getting infected by a bacteriophage, they can actually connect together on their own through something called the 6 pili or a pilus which connects the two bacteria and allows a plasmid to be shunted or right from one directly into the other. Another way to share genetic information between bacteria. And this we think is a good part of the way that antibiotic resistance, besides just being replicated by bacteriophages, um, can be passed on to other bacteria. So our plasmids carry genes for antibiotic resistance. Um, and as we know, antibiotics kill bacteria that are sensitive to them, but bacteria that have certain R plasmids are resistant to those to certain antibiotics. So either through natural selection, killing off the ones that don't have it, so only the ones that have that gene survive and pass it on, and via conjugation or transformation, we can get we can spread that to other bacteria, and that's one way we think that antibiotic resistance spreads. Another thing that can happen to increase variation, not necessarily always in reproduction, is basically crossing over, but it doesn't have to happen during meiosis, like we talked about before. We call it transposition, the movement of DNA segments within and between DNA molecules. So in crossing over, we did it between homologous chromosomes, right? Between the moms and the dad's chromosome number one. So it'd be the same genes on the same chromosomes, just switch which origin chromosome they came on. Transposition is basically the same thing, except between different non-homologous chromosomes. So we can see these are definitely non-homologous chromosomes here. We got a long blue one and a short orange one, but we can still switch those there. So it increases genetic variation in the same way that crossing over does. In fact, even perhaps more so. Still got all the genetic information, but they're in different chromosomes. So when you go to replicate the cell, you're going to have some differences. One other one to talk about is viral recombination. And this we think is maybe somewhat how we get new strains of viruses that combine some of the functions of two different ones. So related viruses can combine or recombine genetic information if they infect the same host cell at the same time. So we got this blue one and this red one. If they get in there at the same time, they actually might wind up combining their genetic material to make a new hybrid strain that has, well, here we got spike proteins from one and the other. So they're able to infect a broader range of hosts, perhaps, or get into hosts easier, all sorts of things. This may be what's happening with some of the new COVID strains. So, how does chromosome number affect these things? So, alterations of chromosome number or structure can cause some genetic disorders. Large scale chromosomal alterations in humans and other mammals often leads to spontaneous abortions or miscarriages. If you got large problems like you don't have enough chromosomes, or you have too many chromosomes for some chromosomes, or other major troubles, the developing zygote is going to get spontaneously aborted pretty early on. In fact, many more women are pregnant, have been pregnant, than realize it, because what winds up happening is they get some implantation, um, but it doesn't stick around very long. And so they wind up having just a slightly late and slightly heavier flow than normal, can be a sign of something like this happening. Plants, on the other hand, seem to tolerate those genetic changes better than animals do, and I'm not sure why. Abnormal chromosome number, we've talked about this a bit before with non-disjunction, which I've got over here. Non-disjunction is when, for some reason, they don't split right up here in meiosis one. And when you're done, what winds up happening is you wind up with some of your cells having too many, some of your cells not having enough, all sorts of different things. So aneuploidy is what we call the result of fertilization of gametes in which non-disjunction occurred. Offspring with this condition have an abnormal number of a particular chromosome. Um, More often than not, it's a spontaneous abortion, but there are some that survive. A monosomatic zygote has only one copy of a particular chromosome. A trisomatic zygote has three copies. Polyploidy, like triploidy, like we talked about in the last one, is a condition in which an organism has more than two complete sets of a particular chromosome. You can get one extra one, or you can get four even. Polyploidy is common in plants, but not animals. Um, they are, however, in mammals more normal in appearance. You can't see it visually as clearly as you can with monoploidy. or aneuploidy. Alterations in chromosome structure could also have some effect. So the breakage of chromosomes can lead to four types of changes in chromosome structure. One, a deletion, removes a segment. Boom. So you no longer have whatever genes happen to be in that section. You're not going to be able to produce. Hopefully, you still produce them on the other chromosome that you got from the other parent. Um, so it's kind of like having, you know, monosomy for those sets of genes. Duplication, you wind up with extra copies of that gene, so you'll wind up with more expression of that particular polypeptide or RNA. You can have inversion, where we just kind of flip the order of them, and we can have translocation, which we talked about before, where we can move it from one chromosome to another. Now, chromosomal alterations can lead to human disorders, and some are very serious. Sometimes aneuploidy appear to upset the genetic balance less than others, resulting in individuals surviving to birth and then beyond. Like we said, most of the time they don't survive to birth, but there are some that do. These surviving individuals have a set of symptoms, which we call a syndrome, which is characteristic of that type of aneuploidy. Probably the most common one people are familiar with would be Down syndrome or trisomy 21, which results from three copies of the 21st chromosome. Right now, it affects about 1 in 700 births in the United States per year, or 1 out of every 700 births um, in the United States per year. It does increase in frequency with the age of the mother. Um, as of this moment in time, I am not sure that anybody has come up with a satisfactory explanation for that that's widely accepted. Um, I may be wrong. I try to stay up to date on things, but I'm not a geneticist. Aneuploidy of sex chromosomes. So having a different number of sex chromosomes than normal is as opposed to just the regular autosomes like we were talking before, can produce a number of conditions. You can get three X's, in which case the females are healthy with no unusual physical features. Klinefelter syndrome, which is XXY. So you have two X's and then a Y. Monosomy X, otherwise called Turner syndrome, produces X0 females, just one X. They are generally sterile, and it is the only known valuable monosomy in humans. And Turner syndrome is the only one of these that was specifically mentioned in the AP Bio curriculum. The other ones, I don't think you need to remember which ones are which, just that it's aneuploidy. But Turner syndrome, you may need to know that one. All right, join me in the next section when we talk about biotech, guys.